is Vatsala Selvam in the Division of Lifecycle API. I'm going to answer some of the very good questions that we received on topics such as DMF Form 3938, Agent Appointment, Letter of Authorization, Pre-assigned DMF number, Annual Report, and Communication. The first question asks, should we submit an administrative information page with every submission when the DMF Form 3938 is submitted? When the DMF Form 3938 becomes available for use, no need to submit an administrative information page with every submission, since a form should have the information that an administrative page would have. If an agent is appointed for a DMF, please make sure the holder signed agent appointment letter is submitted to the DMF. Just mentioning in the form is not enough. If any addition, change, or deletion of holder, manufacturer, and agent information, providing the updated information in the form is not sufficient. Need to submit an amendment to the DMF reporting the change. The next question is on agent appointment. Is an agent appointment letter mandatory for a secondary DMF? There is no regulatory requirement to appoint an agent for a DMF. If the holder decides to appoint an agent to submit and receive communications for either a primary or a secondary DMF, then an agent appointment letter should be submitted to each DMF separately. The next question is on letter of authorization. Should the secondary DMF holder include the site details in the letter of authorization? In cases where all of the facilities listed in the secondary DMF are not being used to support the primary DMF, we recommend that the specific sites supporting the primary DMF be included in the letter of authorization to provide clarity. Here is another question on LOA. What is the correct procedure for the withdrawal of a specific LOA in ECTD? Remove the LOA and then add the withdrawal letter, or replace the LOA with the withdrawal letter? The best practice would be to submit withdrawal of letter of authorization using the replace option in ECTD to withdraw the letter of authorization that was already submitted to the DMF. The next question is on a pre-assigned number. How long is a pre-assigned DMF number valid? A pre-assigned DMF number does not expire. If there is any change to the information provided in the request for a pre-assigned number, send an email to dmfquestion@fda.hhs.gov requesting to cancel the assigned number and submit a new request for a new pre-assigned number. If it has been over 3 or more years since you received the pre-assigned number, we recommend to send an email to DMF Question mailbox asking for status. We can verify and let you know if it is okay to use the same pre-assigned number. The next question is on annual report. Is there a specific amount of time that should be observed in between an amendment and an annual report submission? No, there is no time duration to be observed in between an amendment and an annual report submission. Any new change, addition, or deletion of information made to the DMF should be reported only by an amendment as they occur. Quality information should not be included in the annual report. Question: if there is no change to report, is it necessary to send an amendment every year solely to update the long-term stability data, or it could be submitted the following year? There is no requirement to submit stability data annually, but if the stability data has been updated, it should be submitted to the DMF as an amendment. The annual report needs to be submitted to the DMF every year in order to keep the DMF status active. Please check the webinar and administrative aspects of managing a DMF for information to include it in an annual report. Here is the last question. Would an ANDA holder who references a DMF be notified if there is an issue with the DMF, such as missed annual report? Yes, an ANDA holder who references a DMF is notified of any letter of authorization issue, user fee status, review and administrative status of a DMF. Thank you and have a nice day.
Hi, I'm Jonathan Resnick, and we'll be answering a few questions on electronic submissions. Question: should cover letters and forms replace previous version, or should they always be new? Cover letters and FDA forms under headings 1.1 and 1.2 should use an ECTD lifecycle operator of new, since they describe the contents of the sequence. Including a descriptive leaf title, which includes the date of the submission, can be helpful to the reviewer. Question: do we need to submit ECTD for a legacy product which has been submitted or which has been on the market for a long time? The requirement for submissions to DMFs to be an ECTD is not based on the age of the DMF or product. All submissions to DMFs, even if the DMF was originally submitted before the requirement went into effect, must be an ECTD. ECTD requirement applies to Type 2, 4, and 5 DMFs, and submissions to Type 3 DMFs can be submitted in ECTD or non-ECTD. We recommend checking the FDA guidance on ECTD submission requirements for additional detail and look at some scenarios where a waiver of the ECTD requirement might be granted. In the bullet below, we have a hyperlink to the formal title of the ECTD guidance. Question: for letters of authorization that were previously submitted as paper format, can the withdrawals of those LOAs be submitted with cover letter section 1.2, since there is nothing to replace in section 1.4.1? The draft DMF guidance goes into detail about this, and it really gives a lot of good information about where to place content regarding a withdrawn LOA within the ECTD. We have a couple of excerpts from the guidance here, which may help with the answer to this question. The first excerpt says, in ECTD section 1.4.3, DMFs must list each party currently authorized to incorporate by reference any information in the DMF. The list should only contain authorized parties for which LOAs have been submitted and should be updated whenever a new LOA is submitted or an authorized party is withdrawn. To withdraw authorization, DMF holders should submit a withdrawal of authorization letter to the DMF and notify the authorized party. The withdrawal letter should replace the LOA in ECTD section 1.4.1. So that withdrawal letter should go into section 1.4.1. If the original LOA was in paper, you can still submit that withdrawal letter in section 1.4.1.
Hi, I'm Commander Hannah Pham, along with Lieutenant Commander Evelyn Hong. We'll be answering some of the great questions we received from our presentation on drug master files from a GDFA 2 user fee perspective. The first question asks, does a facility manufacturing only API intermediates need to pay a facility fee? And which facility fee does an FDF intermediate manufacturer need to pay when it is identified in an approved ANDA? A facility that manufactures only API intermediates is not subject to GDFA facility fees. However, a facility manufacturing FDF intermediates or in-process FDF mixtures is subject to an FDF facility fee or a CMO facility fee if it meets the criteria to be a CMO, when that facility is identified in an approved generic drug submission. The second question asks, if a new API facility is listed in a pending ANDA, is it required to pay the API facility fee? And when the ANDA is approved in mid-fiscal year, which fiscal year is the facility required to submit the payment? The new API facility in question is not subject to an annual facility fee until the reference in ANDA becomes approved. And if that ANDA in question is approved in the middle of a fiscal year, the new API facility is not required to pay for the facility fee for that fiscal year. Instead, the facility will incur the API facility fee for the first time in the next fiscal year. And at this time, I'd like to pass this on to Lieutenant Commander Hong to address the remaining questions.
Thank you, Commander Pham. Next question asks, if secondary DMF is referred in a primary DMF, that is, DMF of API base is referred in a primary DMF of API sold, when API base is not being directly used in the finished product's manufacturing. Is the DMF fee required for the secondary DMF of API base? Similarly, is the DMF fee applicable for the Type 2 DMF intermediate? The secondary DMF and the API DMF intermediate are not subject to the DMF fee. Only the primary DMF is subject to the DMF fee if referenced in a generic drug submission. Our last question asks, if the DMF fee is paid for a Type 2 DMF, does it need to submit another DMF payment for adding a new API facility to replace an existing API facility for that DMF? The answer is no. The DMF fee is a one-time fee, in which once the fee is paid, no additional payment is required for adding a new API facility.
Hi, I'm Erin Skoda, and I'll be answering a few questions that we received via email on review timelines, referencing applications, and communication. The first question has to do with review timelines and communication. If the review timeline also depends on the referencing applications, how do I, as a DMF holder, know where we are in the review process? In general, you can tell where you are in the review process by staying in constant communication with each of your referencing applicants. In my full talk at the DMF and Drug Substances workshop in early March, I presented this timeline. And these points in the review process that are noted in red are our suggested communication points between the DMF holder and the ANDA applicant. And these apply to each of your referencing applicants. So specifically, if you communicate right after the DMF fee is paid and the DMF is submitted, following ANDA receipt, following the ANDA filing decision, also when you respond to the DMF first letter that you receive, and then also when your DMF is adequate, these communication points would be a good minimum to start by so that you can be in constant communication and know where your applicant is and where you are in the review process. And again, this applies to each of the referencing applicants. These are just the minimum recommendations that we have, and additional communication points would be even more beneficial. The next question is also related to referencing applications. And the question is, what is the impact when a DMF is referenced by more than one ANDA? What if the applications are for different dosage forms or indications? We consider that the DMF as it supports each ANDA. It could, for example, be adequate to support one ANDA, but inadequate to support a different ANDA. Some things to consider when your DMF is referenced by more than one application are the following. If the new drug product has a different maximum daily dose as indicated on the drug product label, this may affect ICH-Q3A and ICH-M7 impurity limits. So you should consider those in your drug substance. If your drug product has a different duration of use, this may affect your ICH-M7 impurity calculations. And then finally, if your drug product has a different dosage form and or route of administration, this may require different physical properties or characterization data, such as the solid state form. The next question is related to review timelines. During the ANDA review cycle, can a DMF holder delay submission of an annual report? Will submission of an annual report impact the ANDA review timeline? We do not recommend delaying the submission of annual reports. Only quality amendments that are received late in the review cycle may impact the application's timeline. Annual reports should not contain any quality information, and if they do, when we receive them, we recategorize them as quality amendments. However, sometimes we may miss this recategorization, and so if you miscategorize a submission, that may further affect our review timelines. The next question deals with communication, and the question is, where can we find a list of FDA's common acronyms? You may find acronyms defined throughout our recorded presentations, but if you still have additional questions regarding FDA's acronyms, of which we realize there are many, the agency has a searchable list of acronyms at the link below. Thank you.
Hi, my name is Jayani Pereira, and thank you for submitting the questions for the follow-on webinar. I'm going to answer a few questions received on the topics of completeness assessment and timely consult and early information request. The first question is related to completeness assessments and asks, can FDA expedite completeness assessment review if requested by the holder to reduce the likelihood of a refuse to receive action on the referencing Amda? Yes, FDA will consider requests to expedite the completeness assessment from the holder or an authorized party. Requests should be sent to DMF OGD mailbox and should include the following information: DMF number and DMF fee payment date, and estimated time frame for referencing application submission. Note that industry does very well in submitting DMF fee payments early, such that expedite requests for CA are rarely needed. Continued strong performance in this area allows us to consider CA expedite request when circumstances warrant. FDA will internally expedite ACA under the following circumstances: when any error was made on the part of FDA, or when a referencing application is submitted and there is an open CA cycle on the DMF. This question is also related to completeness assessments and asks, if an FEA number is not available for a manufacturing, testing, or an advanced intermediate manufacturing facility, does it impact the application filing status or the completeness assessment outcome of a Type 2 DMF? The completeness assessment for Type 2 API DMFs under GDUFA Guidance for Industry, item number 16 states, central file number, facility establishment identifier, and data universal numbering system numbers should be provided if available. If the FEI number for manufacturing, additional processing, release and stability testing, or critical intermediate facility is missing from the submission, an incomplete comment will be issued. This can only impact the filing of an application if it is not resolved prior to the filing decision. This is the last question related to completeness assessments and asks, when does the completeness assessment review of the DMF start? Is it only after 6 months from the submission date of the DMF? The completeness assessment review starts when the fee status is met and the DMF is active. DMF active means the DMF documentation has been submitted and an acknowledgement letter has been issued. As stated in the GDUFA 2 commitment letter, the agency will strive to complete the initial completeness assessment review of 90% of Type 2 API DMFs within 60 days of the later of the date of DMF submission or DMF fee payment. For details, please refer to the poster presentation on completeness assessments. The next couple of questions are related to timely consult and early information requests. The first question asks, with new knowledge gained, starting material designation that was acceptable in the past may not be acceptable now. You mentioned, and I paraphrase, that TCIR starting merit evaluation only applies to DMFs that have never been reviewed. Does this prevent FDA from revisiting and re-evaluating DMFs that have been reviewed before regarding their starting material designations? During the TCIR process, starting material evaluation is done only for original DMFs which have never been reviewed. Proposed starting material is evaluated using principles outlined in ICH Q11 guidelines. For previously adequate DMFs, the starting material will be re-evaluated during full scientific review process only if significant changes are made to the manufacturing process or the starting material has been redesignated. Again, the ICH Q11 guidelines will be followed. The last question related to TCIR and asks, if the DMF holder would like to update the DMF with newly added facilities during the referencing and the review cycle, when is the best time for the DMF holder to submit the information to the FDA? Can DMF holder file unsolicited amendment to the DMF? Does it impact approval of the referencing ANDA? There are three scenarios. If the newly added facility supports the referencing ANDA, then the DMF holder should notify the agency in an unsolicited amendment so that the information about the newly added facility can be relayed to the Office of Pharmaceutical Manufacturing Assessment. If the newly added facility needs to be evaluated and an inspection is needed, then the performance goal date may need to be extended to allow the inspection of the facility before approval. If the newly added facility is not supporting the referencing application, then reissue the LOA specifically listing the DMF facilities that do support the application. If the newly added facility doesn't support this or other applications now, but will be in the future, then the DMF holder should consider adding the facility post-approval and delaying submission to the DMF until that time. Thank you.
Hi, this is Commander Dave Skanke, and I'm going to answer some of the very good questions that we received on topics such as GDUFA, some general DMF issues, and communications. The first question asks, you mentioned that when an API excipient mix is for stability purposes, only an API facility fee will need to be paid. Are we talking about physical as well as chemical stability, or only chemical stability, which is mentioned in the API definition in GDUFA? The definition of active pharmaceutical ingredient under GDUFA for the purpose of determining facility fees includes the following. A substance or a mixture when the substance is unstable or cannot be transported on its own. The FDA has interpreted unstable to refer to both the chemical and physical stability of the substance. Please refer to the draft FDA guidance for industry assessing user fees under the Generic Drug User Fee Amendments of 2017. When claiming that the mixture is made for the purpose of mitigating a stability issue and thus qualifying the facility for the GDUFA API facility fee, supporting information such as data and literature references should be provided in the DMFs. This question is about facility fees for intermediates. Do facilities manufacturing API intermediates have to pay the GDUFA facility fee, or is it just the final API facility that is required to pay? It is the nature of the API intermediate that determines whether or not the intermediate facility is subject to the API facility fee. In practice, most API intermediate facilities will not incur the fee. GDUFA includes the following statement in its definition of API for determining facility fees. An API includes a substance intended for final crystallization, purification, or salt formation, or any combination of those activities. Let's apply this definition to the scenario below to see which facilities will incur fees. Here we have the regulatory starting materials A and B going through multiple steps across 3 different facilities to the final drug substance G. Intermediate D, produced at Facility 1, would not incur a fee. Intermediate D is one or more chemical transformations removed from the final API and does not meet the GDUFA API definition. Facility 2, on the other hand, which produces intermediate F, which is the drug substance base, would incur the facility fee. This is because the intermediate F meets the GDUFA API definition because a salt formation step produces the final API. Facility 3, of course, would also incur the fee because that is the final API site. This question is about drug substance deficiencies in application CR letters. Please explain why sometimes DMF-related deficiencies are issued to the ANDA application and not the DMF. FDA realizes that this situation has been a cause for confusion to industry in some cases. It is caused by the following elements of the review process. Different reviewers are responsible for the API-related information in the application and the DMF. Reviews were largely not synchronized in time. Lack of coordination and communication between the DMF and the application reviewers. FDA is addressing these issues through the implementation of the Integrated Quality Assessment Aligned Teams Initiative, which started in August of 2020. This should reduce inconsistencies in the content and timing of application and DMF letters. Reviewers from different disciplines work together on multiple projects and thus are more familiar with each other. This should improve synchronization of the reviews by members of the team and improve the consistency of communications between members of the team. Please refer to poster number 10 on ANDA Aligned Teams by Steve Kinsley and Wei Song for details on the IQA Aligned Team process. This question is about available for reference requirements for DMF referenced in prior approval supplements. Could you please clarify whether the DMF must be listed in the DMF available for reference list when we are filing a PAS for a new strength addition to the approved ANDA using the same DMF? That is, no change in the API source. If the Drug Master File was appropriately referenced in that application by a letter of authorization prior to the start of GDUFA on October 1st, 2012, then there is no requirement that the DMF pay the fee or be on the available for reference list to support that prior approval supplement for that application. If the DMF were being referenced for the first time by that application in the recently submitted prior approval supplement, then the available for reference requirements would apply. Note that when the Drug Master File referenced in a PAS must meet available for reference requirements, that the FDA can do the following. For supplements that are subject to a filing review, such as a new strength supplement here, we could refuse to receive that supplement. For supplements that are not subject to a filing review, such as for an alternate API source, we can suspend review on that supplement until the available for reference requirements are met. This question is about unsolicited amendments to DMFs. Often the ANDA under review is not the only customer referencing a DMF. It is difficult for the DMF holders to provide adequate and timely continued support to all customers if they are concerned about an extended timeline due to what may be a very simple unsolicited submission. How can industry effectively deal with these situations, especially for older DMFs that are multiply referenced by approved products? FDA does understand that these situations can and do arise, and we have the following recommendations. Make sure you are aware of the action dates for the open referencing applications by getting that information from your customers. Let your customers know you are planning an unsolicited amendment to the DMF and your proposed timing so that they are aware. Reach out to us at DMF OGD to seek further advice if needed, noting that we cannot discuss application goal dates with you. Understand the consequences, i.e. goal date extensions, when an unsolicited amendment must be submitted. Note that in our experience, most, but not all, unsolicited amendments can come in with timing that does not adversely impact an application timeline. In some cases, it may be unavoidable, and the DMF holder must make a business decision that won't be completely satisfactory to all customers. This question is about unsolicited amendments to DMFs. Are annual reports and letters of authorization considered unsolicited amendments that could adversely impact an application timeline and delay an approval? Annual reports and letters of authorization are administrative amendments and are not treated as unsolicited amendments in the same way as amendments that contain quality information. Administrative amendments do not trigger a review and cannot result in goal date extensions or deferrals, nor can they impact the adequate or inadequate status of the DMF. Other types of common administrative amendments are change in holder, change in subject, new U.S. agent appointment letters. The only administrative amendments that can impact an application are an LOA withdrawal or a DMF closure request. Note that annual reports should not contain any quality information, including updated stability data. If they do, they will be treated as both an annual report and a quality amendment.
Hi, this is Brian Connell, and I'm going to answer some questions regarding a post-approval changes to drug substances draft guidance. A change in the manufacturing process of a drug substance results in the appearance of new process impurity X in an intermediate, and therefore there is a new non-equivalent impurity profile when compared to the pre-change intermediate. A drug substance manufacturer adds a test to control this process impurity at its point of origin and demonstrates that impurity X and its potential downstream analogs are properly controlled based on data generated for a risk assessment, considering ICH Q3A, Q3C, Q3D, and M7 guidelines. Even though the impurity profile is not exactly the same before and after the change, is this approach acceptable? If acceptable, how much method validation data should be submitted? Pre- and post-change impurity profiles are not required to be equivalent, so yes, this approach is acceptable. Because of the manufacturing change, the acceptance criteria for the new impurity X in the intermediate specification should be justified based on a risk assessment that includes a hazard classification per Section 6 of ICH M7. ICH M7 Section 8.1 discusses possible control strategies for process impurities. The updated control strategy should address downstream impurity analogs if necessary. The agency considers this necessary when the new impurity X is controlled in the intermediate at a level higher than the appropriate drug substance limit for impurity X and its potential downstream analogs based on either ICH Q3A or M7 as applicable. Support for a control strategy could include results of a spike purge study, a calculated purge factor analysis, a demonstration of downstream detection and quantitation of the relevant impurities by the routinely used impurities methods, or some combination of all of these approaches. Analytical methods should be developed with sufficient specificity and sensitivity for their intended use. Unless the routine drug substance methods are being updated, full method validation does not need to be submitted. However, we generally do recommend that some basic method information is provided, including a basic method description and a statement of the limits of detection and quantitation. The question is, our product was approved prior to the adoption of the ICH M7 mutagenic impurities guidance, and a risk assessment for potentially mutagenic impurities was not performed at the time of approval. We are transferring the drug substance manufacturing process to a new supplier, but no changes have been made to the manufacturing process, reagents, or specifications. Do we need to conduct a risk assessment now? Generally, no. A new risk assessment is not necessary if the supplier change is the only change. Per ICH M7 Section 4.1, a change in the site of manufacture of the drug substance intermediates or starting materials, or changing raw material suppliers, will not require a reassessment of mutagenic impurity risk. However, a risk assessment may be necessary if the change falls into the scope of ICH M7 Section 2. Specifically, if there is a change made in indication or dosing regimen which significantly affects the acceptable cancer risk level, ICH M7 will apply. For example, if the new supplier is added due to market demand for a new referencing product with the same drug substance but with a higher maximum daily dose than the originally approved product, a risk assessment would be necessary. Note that a risk assessment would be necessary in this example even if a new supplier was not added due to the increase in the maximum daily dose of a referencing product. The question is, do the requirements and recommendations in the post-approval changes to drug substances draft guidance apply to drug substances used in combination products? Yes. The recommendations in the guidance apply to multi-API drug products and each drug substance therein. For example, if the drug product is an oral tablet containing both APIs A and B, the guidance applies to each API, A and B, separately, and the combination final drug product containing APIs A and B. The next question is, if a change is made and there is a new impurity in an intermediate, and the impurity, as well as the possible downstream analogs formed from this impurity, are shown to be absent in 3 batches of drug substance, would this be sufficient to avoid updating the specifications of the intermediates and the drug substance? A central principle of the draft guidance is that the manufacturing process can be adequately assessed by comparing 3 consecutive batches of pre- and post-modification material. If the required ICH M7 and Q3D risk assessments have been completed, and the new impurity and its potential analogs are absent from the drug substance based on application of the appropriate threshold limits, then yes, the drug substance impurity profiles, pre- and post-change, would be considered to be equivalent, and no specification change to the intermediates or the drug substance would be required.
Hi, this is Cassandra Abelard, and I'm going to answer some of the questions that we received on hidden facilities. Question: does the information for the QA contact at the site need to be provided in the DS section, or does that information belong on the Form 356H? The answer: the information for the QA contact at the site should be provided in the drug substance section as well as on the 356H if the QA contact is the contact person for the site. If the contact person for the site is an operations person or a president, then that person's information should be included on the 356H. In the FDA Guidance for Industry, Identification of Manufacturing Establishments and Applications submitted to CDER and CDER, Questions and Answers, it has the following example table for this information to be provided. Note that this table should follow a table of facility information. Question: do the following facilities need to be included in S21 and/or on the 356H form? Pilot scale API production sites, one-time testing sites, drug substance stability storage sites, drug substance storage sites, raw material testing sites, critical intermediate sites. So the 356H should include all the drug substance and drug product manufacturing and testing sites, as well as critical intermediate manufacturing and testing sites. In general, any site performing commercial manufacturing of your product should be listed on the Form 356H. Again, referring to the guidance for industry, there's some specifications in there about what should be included in Module 3. Module 3 should contain all facilities listed on the Form 356H, as well as R&D sites and any sites that generated data in support of the application. This would include one-time test sites. It should also contain any testing labs that perform functions integral to the control strategy. This would include any test sites that generate release data, stability testing to support the application, as well as analytical development sites. All manufacturing and control sites should be either in the drug substance or the drug product sections of Module 3. If you're not sure if a site should be listed, just include it in Module 3. Question: can you give an example of what should be listed for responsibilities of each facility in S2.1 for firms that perform multiple operations or single tests? The guidance for industry identification of manufacturing establishments in the application submitted to CBER and CDER Q&A recommends the following. Clearly identify all facilities associated with your application in table format at the beginning of the relevant section of Module 3. Include the summary table at the beginning of each relevant section of Module 2. Include the full establishment name and establishment address where the manufacturing function is being performed. Include the FEI number and specific manufacturing operations and responsibilities for each facility, including the type of testing and the DMF number if applicable. Provide the name and title of an on-site contact person, including their phone, fax numbers, and email address. For testing sites in particular, please state the specific tests being performed at those sites. If you decide to use a table format, the following is recommended. Again, that can be found in the guidance document. Question: do hidden facilities apply to NDA DMFs also? Yes, hidden facilities apply to NDA DMFs.
Hello, my name is Wei Liu. I'm going to answer the questions from the presentation of hidden facilities and critical intermediates. The question is, should DMF holder update section 2.1 to include the size of the secondary DMF? The short answer is yes. On slide 16 of previous presentation, we mentioned the determination of critical intermediate is based on complete manufacturing process from the regulatory study material to the final drug substance. So if a secondary DMF is referenced for an intermediate, you are expected to list the secondary DMF number and its manufacturing site information as appropriate in section 2.1 in the primary DMF. Please also provide the LOA for the secondary DMF in section 2.1 or 2.3 in the primary DMF, and clearly indicate which intermediate is manufactured at this intermediate site. As Kathy mentioned in her presentation just now, this information is expected in the Module 3 of ANDA submission as well as ANDA 356H form according to FDA guidance. So please communicate this information to each ANDA applicant which reference your DMF.
Good afternoon, everyone. I really appreciate you all taking time to submit your questions for this follow-up. Your tremendous response suggests how important this topic is, really. While I may not be able to answer every single question in the allocated time, I've chosen them in a way to ensure they cover a variety of topics. Next question. Can I remove an API site from my application if it is OAI and substitute it with another site? If so, what should I consider? Another great question. Yes, you can. You can substitute an API facility. We don't dictate which API facility you should use, but what we recommend that you engage about it with the office that is handling your application. In addition to that, from CGMP perspective, evaluate CGMP deviations noted at your API supplier that is OAI, and that from that you now want to move on, that how it affected, if it did, on the data you have submitted in your application. For example, if data integrity violations were identified at the API supplier, did it affect the batches that you may have used for the biostudies or the exhibit batches? Keep in mind that new facility may still need a pre-approval inspection. So another great question, that what can trigger API inspection? I'm going to take this at a higher level approach, because basically what can trigger an inspection from FDA, whether it's an API or any other type of facility. So FDA conducts many kinds of inspections. Surveillance inspections to monitor the manufacturing process and periodically the quality of the distributed drugs. FDA uses the findings to evaluate whether a manufacturer is complying with CGMPs. In general, the agency does not announce domestic surveillance inspection to the company in advance, but announces international surveillance inspections. Whether inspections are announced often depends on particular cases and the history of specific facilities. We conduct pre-approval inspection as part of the review of an application to market a new brand for generic drugs. This would include various types of facilities included in the drug product manufacturer, including the API suppliers. And we conduct for-cause inspections when they are triggered by FDA has reasons to believe that a facility has serious manufacturing quality problems, or when FDA wants to evaluate corrections that have been made to address previous violations. For-cause inspection can be announced or unannounced, whether domestic or international, depending on the specific situations. Next question. If API is manufactured by a contract manufacturer, does the contract manufacturer need to validate the API process? Again, I think this goes to the heart of the responsibility of a contract manufacturer in general and as an owner. So I'm going to take it at a higher level approach, and that will give you more than you need for such a situation. So regardless of who manufactures your product or the agreements you have in place, you as a owner are required to ensure that the product that you put in distribution meet predefined specifications prior to distribution, and that it's manufactured in accordance with the FD&C Act. If there are operations that are critical to the purity and quality of the API, then critical parameters should be defined to ensure processes can consistently deliver needed quality. This also involves documented verification that equipment and ancillary systems can perform effectively and reproducibly based on the approved processes, method, and specifications. Everyone involved, contract manufacturer and DMF holder, are responsible to ensure the quality. As an owner of the product, you are responsible for final approval or rejection of the batch, while contract manufacturer may still be responsible for GMP. What is expected that an API, is it expected that an API manufacturer will be inspected every 3 years? Well, there is no set frequency. Before 2012, the Act requires the FDA to inspect domestic drug manufacturing sites every 2 years, and no such time frames were outlined for the foreign sites. The shift to overseas production of U.S. goods, including drugs and their components, predominantly occurred in early 2000 resulting in a large imbalance in which facilities were inspecting. That's when FDACIA changed the requirement for FDA to inspect domestic and foreign drug manufacturing sites in accordance with a risk-based schedule. With the passage of FDACIA, FDA's drug inspection program shifted from one focused heavily on U.S.-based facilities through the early 2000s to a program that since 2015 has conducted more foreign than domestic drug inspections. The site selection model is at the core of FDA's surveillance inspection prioritization program and ensures a uniform approach for domestic and foreign facility inspections. The agency uses the model to calculate score for every facility in its catalog using risk-based factors that I discussed during the webinar. If my facility is on import alert, can I ship API to a manufacturer outside the U.S. to make drugs intended for the U.S. market? No. If your facility is on an import alert, you may not supply drugs directly or indirectly. Now, we occasionally carve out drugs that are medically necessary or in shortage and allow them to be offered for importation, even if manufacturing facility is on an import alert. If this is the case, we clearly outline what drugs may be offered for importation in the communication notifying implementation of an import alert.
Hello, I am Bapu Gadam. I'm going to answer some of the good questions received on regulatory considerations in demonstrating complex API sameness. The question is, what is the definition of complex API? Per GDUFA commitment letter, there are several complex drug products. So one of the classes, the complex drug product is having complex active drug substance. The current thinking is any active substance structure that needs orthogonal characterization methods with product-specific analytical techniques, and also the characterization is dependent on the manufacturing process and functional data are considered as complex API. Are botanical drugs complex APIs? As for the draft guidance for botanical drugs, the drug substance characterization requires the product-specific techniques and method for the manufacturing. However, the complexity is depending on the individual drug substance and or drug product. The guidances and recommendations are applied based on the properties of the individual drug substance and drug product. There are a couple of questions related to this draft guidance on peptide. In the case of peptide with an official USP monograph that is manufactured by recombinant technique and the same peptide submitted by DMF holder is manufactured using a synthetic process, what is FDA's stance on API sameness? Are there any additional requirements or techniques that should be used to demonstrate API sameness? Another question is, for linaclotide being a synthetic peptide, do we need to follow the draft guidance for control of impurities? If the peptide is officially USP and manufactured by recombinant technique and same peptide submitted by DMF holder and it is manufactured using synthetic process, then the DMF holder is advised to follow our recommendations mentioned in our draft guidance. As for our current thinking, we advise you to follow our recommendations mentioned in this draft guidance for industry for the control of related impurities in linaclotide. Please also note that linaclotide has product-specific guidance. Please note the guidance for the recombinant versus synthetic was issued for these 5 peptides listed here. There are 2 questions regarding API sameness. Do the API sameness requirements described in your presentation also apply when qualifying an alternative API supplier for an IR tablet? Does an ANDA applicant need to repeat sameness or comparative studies of drug product with RLD if the stability data of the batches made with the new API is trending similar to batches with approved API? Is similarity in the stability data of batches made with the new API with approved API sufficient to demonstrate that there is no negative impact on drug product quality, safety, potency, and purity? The API sameness should be established for the API obtained from the additional new source or alternate API supplier. The data required depending on the nature of the drug product and drug substance. The stability data is useful to establish the expiry date of the drug product. Stability data of drug product does not establish the sameness of API from the new source.
Hi, this is Lieutenant Suresh Jayasekara. I'm going to answer some of the very good questions that we received related to complex APIs. The question is, for peptide drug substances, is it necessary to perform the bioassay test for characterization purposes? Is a routine bioassay release test complementary to the assay by HPLC required in the drug substance specification? Bioassay test is useful only at the formulated peptide API stage, that is drug product stage. Therefore, in the complex peptide DMF submission, the bioassay test is neither required in the drug substance characterization section nor in the drug substance release specification. The next complex API question reads, another topic that we would like to clarify is peptide sequencing as a proof of primary structure. Is it expected to be demonstrated once in the context of RLD sameness and characterization, or should it be a part of the drug substance specification routine identification test? Peptide sequence analysis is one of the key characterization methods that we have to provide along with other primary structure characterization methods in the section S3.1 of the DMF. It is not required to add the peptide sequence analysis test as a part of routine identification test in the drug substance specification. The next question. Does the agency expect to obtain generic drug characterization in context of aggregation or oligomerization state at API stage in the DMF? If yes, should it be a part of RLD sameness study? The evaluation of higher order structures such as polymer or oligomer aggregation states are meaningful only at the formulated API stage, that is drug product stage. Therefore, higher order structure studies are not required in the DMF submissions.
Hello, this is Keduo Qian, and I'm going to answer some of the questions concerning complex API. The first question reads, for complex APIs, when the dose is very low, how to extract the API from RLD finished dosage form? For example, when the RLD is metal complex, can FDA provide the extraction procedure? The second question reads, what kind of information can we obtain from a pre-ANDA meeting? I will answer both questions together. For the extraction of the complex API from the RLD, please note that the sample preparation and composition strategy is different case by case. The FDA doesn't have a specific recommendation on how to extract the complex APIs in order to establish drug substance sameness. However, the general principle was laid out in the poster. It is up to the applicant to demonstrate a complete and clean recovery so that the analytical results are representative. In some certain circumstances, for example, when the finished dosage form is a relatively simple injection, or the drug substance constitutes absolutely majority of the drug product, a direct head-to-head comparison with the RLD finished dosage form may be acceptable. In other circumstances, the Amda applicant is welcome to submit a pre-Amda meeting package to discuss with the FDA on specific scientific issues or questions. Please note that the FDA expects the prospective applicant has enough knowledge of the complex product to allow FDA to provide appropriate feedback early in the process. For example, the applicant should have generated its own data to be discussed. The next question is, after approval of Amda, if the applicant wants to use an alternate API, does all the characterization done at the time of submission needs to be repeated for the alternate API? Please note the request for repeated test will be different case by case. The DMF of the alternate API should establish the complex API sameness. Depending on the finished dosage form preparation process and its impact on the drug substance structures, some of the sameness tests may need to be repeated. The applicant is welcome to consult with the FDA on specific questions.
Thank you to our presenters for those great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. The first group of questions will be directed to Dr. Skanke. And the first question, how long is a DMF letter of authorization, or LOA, received by an applicant from a DMF holder considered valid? Would it be 10 plus years old letter, for example, be considered valid as long as all the information in the LOA is still correct and current?
Yes, that's a good question. There is no expiration date on an LOA as long as the information in the LOA is still current. For example, if the authorized party were to change ownership or change name, then you would want to issue a new LOA to reflect that change. But as long as the information's valid, the LOA does not expire.
Thank you for responding to that question. The next question for Dr. Skanke. If a different route of sentences replaces the original route of sentences in a DMF in which the DMF fee has already been paid, is a new fee required?
Good question. The DMF fee is a one-time fee, and changes to the drug master file after the fee has been paid do not require a new fee. So a new route of synthesis would not require a new DMF fee. Thanks.
Thank you for responding to that question. We have another question for Dr. Skanke. If a deficiency related to the DMF is addressed to the ANDA holder, should the response be submitted by the DMF holder also?
Yeah, when these instances arise, the best and most efficient way to proceed is to have some communication between the applicant and the DMF holder and coordinate a response such that the response to the question by the applicant is mirrored in an amendment to the DMF, so that the DMF and the application will be consistent with regard to that particular point.
Thank you for responding to that question. We have one last question for Dr. Skanke in this round. Does the ANDA applicant need to obtain a letter of authorization for secondary DMFs, or do letters of authorization for secondary DMFs only need to be issued to the primary DMF holder?
That's a good question. Our expectation is that the secondary DMF will issue their letter of authorization to the primary DMF, and that the primary DMF will then issue their letter of authorization to the applicant. So there would be no need for the applicant to have a letter of authorization from both the primary and the secondary DMF. Thanks.
Thank you for responding to that group of questions. The next group of questions will be directed to Dr. Selvam. And here's the first question. Does the U.S. agent appointment letter to be signed, is it to be signed by the DMF holder?
Yes, DMF holder should sign the agent appointment letter appointing an agent for a DMF.
Thank you for responding to that question. The next question for Dr. Selvam. A firm was previously assigned an FEI and submitted a DMF Type 2. No annual report was submitted. Now, the FEI and DMF can no longer be found on the FDA website. How can I remedy this situation and update the DMF? Should the firm submit a new DMF?
The firm should send an email to dmfquestion@fda.hhs.gov with the DMF number and the holder name. We will check and let them know the DMF status.
Thank you for responding to that question. I have a couple more questions for Dr. Selvam. Here's the question. Can another DMF be filed for the same subject from the same holder?
Good question. Yes, holder can file another DMF for the same subject. When requesting a pre-assigned number for the second DMF, please indicate the reason for the second DMF, such as change in manufacturing process. It is also better if you include the word process 2 or form 2 or some internal code in a bracket followed by the subject in order to differentiate between the two DMFs.
Thank you for responding to that question. The last question for Dr. Selvam. When a new non-U.S. DMF holder has an office in the United States, does the holder need to appoint them as the DMF agent to correspond with the FDA on behalf of their DMF?
If the company name in the United States is same as the DMF holder company name, no need to appoint that person as a DMF agent. He or she in the United States office is considered to be the DMF holder representative, since they are an employee of the holder company. Their information should be included in the submission's administrative information page as a holder representative. No need to appoint them as an U.S. agent for a DMF.
Thank you for responding to that question. The next group of questions is directed to Dr. Liu. And here's the first question. For adequate DMFs, additional sources for the critical intermediates from the new supplier can that be included under the same DMF?
Thank you for the question. For adequate DMFs, additional source of critical intermediate from the new supplier can be included in the same DMF as long as it has the same route of synthesis. Under GDUFA regulation, the DMF may only contain one single drug substance manufacturing process. Here, the single process means the same starting material and the same intermediates, with minor variations being allowed in solvents and raw materials, as long as the type of chemical transformation in each step remains unchanged. So, if additional source of critical intermediate choosing from the new vendor is different, has a different manufacturing process from your existing vendor, is not allowed to be included in the same DMF. But please be reminded this is only the requirement for the GDUFA. Thank you.
Thank you for responding to that question. The final question for Dr. Liu. For semi-synthetic drug substances, will the intermediate acquired by a fermentation process always be considered a critical intermediate?
Thank you for this question. This is a case-by-case decision based on the risk assessment. During the assessment, we will consider all principles we discussed in the presentation in the workshop to make the determination. It will depend on the overall manufacturing process and the control strategies in both fermentation process and its subsequent synthetic step. So if a secondary DMF is referenced under this situation, it means it will depend on the process described in both primary DMF and the secondary DMF. So specifically, we will look at how many steps away from the fermentation intermediate to the final drug substance, how much the structure features introduced from the fermentation intermediate to the final drug substance, and the complexity of the final drug substance, intermediate specifications, and in-process controls. So this is a case-by-case decision, and even for the same fermentation intermediate, it could be non-critical for one manufacturing process, but critical for a different manufacturing process. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Connell. And here's the first question. If a change in the process causes the presence of a new impurity X to appear not only in an intermediate but also in the final API, is this acceptable assuming an updated drug substance specification, or is it required to file a new DMF due to the new impurity in the specification?
There's no requirement to maintain an identical impurity profile throughout the life cycle of a DMF. So even when there is a specification change to the drug substance, there's no need to file a new DMF. The process change and the drug substance specification change can be reported within the existing DMF.
Thank you for responding to that question. The next question for Dr. Connell. For production of an API by fermentation, in which the API, or at least the base molecule, is generated from the fermentation broth, resulting in the final API, or resulting in a base that is then put into a salt form to create the final API, that is, where the molecular structure does not undergo change after fermentation. How do we apply the concept of changes before or after the final intermediate when there is no intermediate, as defined in ICH Q7?
That's a good question. For semi-synthetic drug substances, such as those produced by fermentation, the recommendations in the guidance only apply to any pre- or post-fermentation synthetic manufacturing steps. So they do not apply to the fermentation process step itself. So in the case as described, the fermentation step would not be subject to the guidance. However, the final salt forming step would be subject to the recommendations in the guidance. Thanks.
Thanks for responding to that question. The next question for Dr. Connell. If we perform process validation after what we consider to be a minor change, for example, a starting material vendor change or a minor process change before the final intermediate, do we need to enter all three process validation batches into the stability study, or will one batch be sufficient?
Yeah, that's another good question, and the answer is it depends. According to the guidance, if the impurity profile is equivalent, then yes, one batch would be sufficient. If the impurity profile is not equivalent after the changes, then we would expect or we recommend 3 batches to be entered into the stability study program. Thanks.
Thank you for responding to that question. One last question for Dr. Connell, and here it is. Does the impurity profile need to be compared when the manufacturing process is transferred to a new facility and scaled up there? Does an intermediate need to be filed?
Yeah, good question. I think an amendment does need to be filed because you are reporting, or you should report the change. A change has happened, so an amendment should be filed. If you're scaling up at a new facility, we would consider that to be a multiple change, which is discussed in Section 11 of the guidance. And we would generally consider that to have the potential of having potentially adverse impact on the drug substance impurity profile and potentially its physical properties. So yes, the answer is the impurity profile should be compared when a process is transferred and scaled up at a new process, and that comparison then should be filed as an amendment to the DMF. Thanks.
Thank you for responding to that question. The next group of questions is directed to Dr. Abelard. Here's the first question. How does FDA select intermediate sites to be inspected? If they produce critical intermediates, does FDA audit them?
Thank you for that question. So typically, yes, the FDA will inspect critical intermediate sites. The inspection determination is based on, as anything else, a risk assessment. Once the intermediate is determined to be critical, then typically an inspection would occur at that time.
Thank you for responding to that question. The next question for Dr. Abelard. Is it mandatory to include the information such as address, details in the DMF sections 3.2.S2.1? For example, analytical method validation is performed at an alternate site. Does this site need to be included if it is not being used for routine release and stability testing of the drug substance?
Thank you for that question. That's a great question. Yes, we would expect these sites to be included, but we also ask that you specify that these testing sites are only being used or were only used for method validation and are not intended to be used for testing of routine commercial batch manufacturing.
Thank you for responding to that question. It looks like we've got just a little bit less than 2 minutes left for one more question. And the last question goes to Dr. Abelard. What if an original DMF where one of the proposed manufacturing facilities of a critical intermediate has an open warning letter from FDA and resolution of the warning letter issues is ongoing? Would you recommend that during the submission of the responses to the first CR, the DMF holder removes this critical intermediate manufacturer from the DMF, proposing an alternate manufacturer along with all the required documentation?
That's a great question. So generally, the agency cannot approve an application that references any facility that's not compliant at the time of submission of the application. The agency can evaluate replacement sites as long as the necessary documentation for the new site is provided. We also typically would not advise, be a consultant on whether or not you should replace the intermediate site. We're just going to stress that we can't approve an application that references a facility that's not compliant at the time of submission. Thank you.
Well, that's all the time we have for questions in this session. A huge thank you to our presenters and panelists for answering all the questions that came in. We'll now enter into our break time and return at 2:35 p.m. Eastern Standard Time.